Helicobacter pylori. Let me see if I can move on to next slide, if that is an option for me. Oh, it does. Okay, great. So we have already studied gram-negative rods, and we are talking about intestinal bacteria. And this bacteria, historically, I don't want to spend much time on that, but historically, most of us were taught that gastric ulcers are due to stress, or they still are, but maybe there's an aggravating factor. But just imagine 20 years ago, nobody would believe an Australian physician if he was to say that no, this is from bacteria. And there's a lot of history that goes over there. He used to go to meetings and present his data. People will laugh at him and make fun of him, but he was pretty much determined. It took him like 10 years. And when people realized that many of these kind of findings were weird, so he did like autopsy of people who had died, and then he could possibly cultivate bacteria, which itself was a difficult task. And eventually, over the time, the scientists realized, and they ended giving him a Nobel Prize for that. So you can see that in science, you have to come with a crazy idea. You have to hold on to that idea. You have to prove that idea, and then fight your way up till it goes. And then again, still Nexium, one of the largest selling drugs, just because of the problems that people have, gastritis and gastric ulcer, remains as one of the most important things that we deal as physicians, and you're going to come up. So I want you to understand the pathophysiology of that, and where is it coming. It will be easier for you to deal with a patient, and it's easy for you to deal with many patients. They may have stress ulcers and so on and so forth. So it's a gram-negative rod. It has if you look at the whole group of helicobacters, we do have two types of helicobacters. One are gastric helicobacters. The other are, though uncommon, intestinal helicobacters. And H. pylori is basically a gastric helicobacter, and it has one of the qualities that it produces very high levels of urease. So urease production is typical for gastric helicobacter. And again, if that is the case, remember that is going to be a diagnostic test for us to use. Same story for pathogenesis. You have one big chapter on pathogenesis, bacterial pathogenesis. And I'll just repeat that bacteria have to first colonize, bacteria have to adhere, they have to invade, they have to do many things before they will pose a problem. So especially a bacteria that has to go and pose a problem in an area which has a pH of 2, a concentrated hydrochloric acid. So you can see the challenges that these bacteria may have, but still they succeed. They colonize, not only inflame the gastric mucosa, but destroy the gastric mucosa, and people have gastric ulcers, and people have problems. So as it stands today, the commonest cause of acute and chronic gastritis, and also for peptic ulcers, is H. pylori, Helicobacter pylori. Not only that, also keep in mind, and this again a concept for you, this is one of those bacteria, and we did discuss a little bit in immunology as well. I'm going to spend some time today to recap that this can lead to an immune response, and that can develop into adenocarcinoma. So people who have gastric ulcers, especially for H. pylori, may end up having cancers as well. So that shows it's important. And then again, mucosa-associated lymph tissue MALT, mucosa-associated lymph tissue lymphoma. So that's another cancerous situation that basically is related to this particular bacteria. If you look at the structure of that, I would want you to pay attention that it has multiple sheeted unipolar flagella. So flagella, which is out there, I mean, I myself I keep on preparing for my lecture till the last minute, so I may have uploaded some slide this morning as well. Just want to be update on some of the material which is out there, not for teaching purpose only, but for my own education purposes as well. And I came across one of the idea that I didn't have before, that why do these bacteria have flagella? So flagella is like a propeller. It's like those fins, rear fins of shark and whales and so on and so forth, they can push through water, push through media, and cause more damage. Those of you who are going to come and work with me for my research part, I do separate cells by a technology called microbead technology, which is basically the cells will adhere based upon the charge. So in this case, you can see there is a paramagnetic bead that is allowing for some of the immunological study that these bacteria can cling on to. And this is a scanning electron micrograph. And basically, you can grow them in culture and then see. This particular slide shows you that this bacteria can exist in bacillary rod-like structures as well over here, and also coccoid. So kind of, pleomorphic. This is basically from the gastric mucosa. So you see the gastric mucosa, and there you see a special stain. It's not a Gram staining. It's called Warthin starry silver stain, and you can see a bunch of these bacteria sitting over there. Now you may want to know that this infection remains very common, and our textbook suggests that is particularly common in low socioeconomic class or even developing nation. But I would argue that still people who are in the western part of the world and with the high level of stress and a lot of chronic diseases and so on and so forth, acute and chronic gastritis remains also a big problem. Humans are the primary reservoir. Person-to-person spread is important. That's interesting, because you can spread from one person to another in a fecal-oral route. So this means leftover food or chewed up food or sharing food or passing on in stool, so on and so forth. You can imagine that how does fecal-oral spread would happen. They are present ubiquitous all over the world with no particular seasonal incidence. If you look at their pathogenesis, as I said earlier, very interesting, and again, it's in your interest as well, and I've tried to color it up over here and try to highlight the fact that this bacteria, once it establishes relationship with you, that's going to be a lifelong colonization. So it's really faithful. It's very faithful to you. It's going to go. The only exception, though, is that if you treat it, so if you keep yourself untreated, it's going to be with you. You have to treat. And there are many multiple factors that are associated for the ability of this bacteria to cause disease. It can colonize, inflame, it can alter gastric acid production, and then again, it will cause tissue destruction. Remember, disease means tissue destruction. Some of the diseases are caused by microbial factors, where microbes themselves will multiply, release toxin, release enzymes, and some of the diseases that we get is because of our immune response. So immune response itself, goes out of control, and I usually say friendly fire, but it's more than friendly fire. Now, the question you will ask yourself, and I will ask you as well, that what causes initial colonization in an area which has a high acid production? So bacteria is clever. Firstly, it will come and close these taps for acid production by a protein which is bacterial acid inhibitory protein. So bacteria will come, release a protein, stop the production of acid, and then also it will neutralize the gastric acid by the ammonia, because I just told you that it has this capability of producing urease. So that basically will allow it to produce ammonia as a byproduct, and then ammonia will shut or neutralize acid production. Now, as I said earlier, it struck me today as well that when these when a bacteria is mortal, it has that long flagella. Of course, there's a good purpose for it, and that possibly exists there for this bacteria to find its way to a thick viscous mucus, because mucus is lining those epithelial structure, so things will be swimming on the top unless you kind of dive into that and you basically push yourself and then add and adhere to those cells. And then again, it does have a surface protein which will help the bacteria to evade immune detection. So pretty smart. When we look at the tissue damage, it causes localized tissue damage because the urease byproduct, and these are some of the byproduct. It has enzyme mucinase, phospholipase, and then the most important thing is that it has an activity of vacuolating cytotoxin A, VACA. That happens to be the most important toxin that is produced by this particular bacteria, which basically will allow it to enter into epithelial cells and then change the nature of cell by producing vacuoles. So it produces kind of this vacuoles, that special structure that I'm going to discuss in a while, that will cause local tissue damage. Another important virulence factor is another gene, which is cytotoxin associated gene, CAGA. So VACA and CAGA, that resides on pathogenicity island containing approximately 30 genes. So this is a whole genetic studies because we are still developing drugs, and there are quite a few drugs which are coming up in the market, and that all are related to immune response. The other important thing that these two important genes are encoded by a structure which is a type 4 secretory system. So what happened in type 4 secretory system is that it's like a syringe. They basically inject those toxin proteins into host epithelial cell. And then again, if there was a structure element of the cell, cytoskeleton, it will inject those toxins into that and basically damage that. So normal like a scaffolding. So it's like you have a scaffolding and it goes over there and shatters that scaffolding. And not only that, also keep in mind your basic immunology that when in tissue damage IL-8 is released, then again it's going to be a chemotactic factor for neutrophils. Neutrophils are good to help you, but in this case, in an effort to help you out, they may also pose a problem, create too much of noise, too much of friendly fire, and causing damage. Then again, neutrophils will come. Remember, this is as I said, that many of diseases are produced by your whole host immune response. So in this case, your own neutrophils, your own IL-8 are accumulating in gastric mucosa, and they are causing gastritis and even gastric ulcer. I just borrowed 2 slides from an Australian physician. Actually, there were 2 of them. One was a physician. And this is from the presentation that he had in Geneva, probably, if that is the right place they give the Nobel Prize to. And you can see this is what he presented. Gastric mucosa, your basic immunology, basic physiology. There's a mucus layer on the top, and there sit Helicobacter pylori with this kind of multiple sheathed flagella. And you can see other cut section over here. This is a human stomach. And you can see a Helicobacter pylori curve gram negative. And basically it has adapted to the local ecosystem. And it has clung to adhere to those villi. And then we need to find out how does it go into those epithelial cells. Again, this is from Nobel Prize org. They do presentation before they give them Nobel Prize. So this is like a PowerPoint from there. You can see infection. This is a particular area, 8 antrum, with most of the time you see, and you can see tons of Helicobacter pylori there. It causes local inflammation. You can see gastritis, and then eventually gastritis will be out of control, and you may have a chronic inflammation leading to ulcer. So ulcer can happen even in duodenal ulcer as well. So you can see a gastric ulcer usually at the lesser curvature, and a duodenal ulcer at the beginning of duodenum. So this is like a basic pathophysiology that you normally see. And sometimes, remember, these ulcers bleed as well. They perforate as well. So that's a life-threatening situation. Now, if you look at the whole pathogenesis picture over here, again, your basic pathophysiology concept applied to this disease process. So you can see this is a host mucosa, right? And this is the host itself, like you. And this is the environment. Environment basically means the cavity of the stomach, whatever you're eating. So what you see over here is that there could be anything. There could be anything in that cavity. You can have, let's say, different type of things in there, like microbes. Remember we talked about that earlier. One such environment is that you have those H. pylori, and these are those specific genes. There are tons of them, but I told you about 2 important genes that they are there, and they will play an important role for allowing this bacteria to penetrate and cause damage, because by the end of the day, host mucosa is the one where damage is showed, where the ulcer actually grows. So there are some elements that will cause a compromise in the whole sequence. Remember pathogenesis, this means the story. How does it start from colonization to disease until you get a robust picture of a clinically acceptable, gastric ulcer and so on and so forth. So you can see this is a normal gastric mucosa, and then Helicobacter will come and affect here and cause superficial gastritis. So the moment it enters over here, your immune system will respond. You will produce all those inflammatory cytokines that you normally do. You will also recruit inflammatory innate immunity cells like neutrophils and macrophages. So they will all come. And then again, if you do not check that initial inflammation over here, it will become chronic inflammation. It will lead to atrophic gastritis. You may have intestinal metaplasia, change in the metabolism of the structure of the cell. And then again, metaplasia, dysplasia, and carcinoma. These are the sequence where epithelial cells go from normal to cancerous. Again, your basic pathophysiology. So you can see from here, many times your own host immune response is going to help you develop these processes. So it is basically a host immune response that is uncontrolled. That's where you need to come in and help your immune system, because you want to get rid of this bacteria over here, and you have to put a stop on bacterial overgrowth. As I said earlier, when it invades, it's going to stay over here. Then some of the risk factor, like if you consume high salt consumption, cigarette smoking, and so forth, these are some of the factors that can aggravate the chronic inflammation or chronic gastritis that you may have in your stomach. So if you're a smoker, right, it's not only going to get heart disease, but it will also mess up your immune system. It will help any chronic inflammation that you have to go out of control and then lead to all these substantial steps. They may end up with cancer. Now, this is something that I said I found out this morning, and I found it very interesting. And you can see that this is a normal epithelial cell. It is covered by gastric mucin, and there's a big gap. So this is like, I would say, like swimming pools, like big swimming pools. If bacteria did not have that kind of propellers, flagella, it would just be floating on the surface. So bacteria has this powerful kind of tool that's going to spin around. These are like cytoskeleton element, and they are controlled regulatory proteins. It's going to spring around, right, and then shoot it into epithelial cell. So that's interesting for these bacteria, that they have this ability to go and cause problems. So if you look at the base of the peptic ulcer, for example, over here, different layers of mucosa, and then again, this is a topmost layer. So epithelial damage, because it creates proteases, and you can see the whole idea is start eroding the mucosal surface till you denude the lower areas like muscle. And if you go a little bit deeper, depending upon how far the crater and the bed of the ulcer is, if you hit the submucosa where the vessels are, the vessel will explode. and you start bleeding. It really bleeds very bad. It can bleed people to death. Or if it keeps on going this way and it keeps on eroding, it will puncture. You get perforation. And just imagine all the acid over there perforates into your abdomen. That's again is a life-threatening situation. So that's at the beginning of the typical gastric ulcer that we have. I would not spend much time on immune response, but I expect that you pretty much already know that. But I'll just recap and give you a little bit of an overview in terms of that specific infection that is over here. Now, again, I would want you to pay attention to the epithelial layer. And then this is thick mucus layer. This is the gastric lumen. And this is all these cells which are basically present in something we call MALT, mucosa-associated lymph tissue. So all those cells that you've known, like dendritic cell, T cells, and so on and so forth. And then again, this is a good key for you to remember if you do not. So H. pylori go over here and they want to pass this barrier. They want to pass this barrier. Once they pass this barrier, then there are cells like macrophages. They want to devour them and take them in. And then antigen processing will take place. These antigen presenting cells will present to Th1 cell if they do. Then again, Th1 type of cytokines will be released. And then you have a typical kind of development of these T cells. T cells are later developed in the gastric lymph nodes. On the other hand, you may have a dendritic cell which has the ability to kind of pass its hand through the epithelial cell and pick up and sample antigen. So that's what it does over here. Sampling of antigen, it picks up an antigen over here, H. pylori, and then presents it to, again, different type of T cell. Now, when it comes through the dendritic cell, you will see, because dendritic cell is a professional antigen-presenting cell, so this may lead into two different type of routes. So you can have a Th1 type and Th2 type of response. So within one immune system or immune challenge, you may get both. And in this case, you can see that you can have Th1 type, making different set of cytokine as compared to Th2 type. So you can see it depends upon the immune response that is created. Now, again, you will see over here, the other important thing that this slide will show you is that once you have a damage over here, you see the damage over here on both sides? If you see the damage over here, and then again, they are special receptors which T cells have, and these receptors will help. And many a time what happens is that, remember I said that sometimes people consider that as an autoimmune disease. So H. pylori will kind of help you develop an autoimmune disease, because the reason is that your cells are making autoantibodies. So you have these autoantibodies over here, and then these cells will bind to those cells, and then we initiate again an immune response where they make tons of those typical autoantibodies, and they keep on attaching to that particular microbe, right? In this scenario, most of the time, you have a dominant pro-inflammatory cytokine, Th1 cell environment, and you can see these autoantibodies being produced. Now, also keep in mind that a typical T helper 1 cell environment, especially in this case, where the Helicobacter tries to get into that, it may develop a tolerance, this particular, to H ions and potassium ATPase, which probably is maintained by T regulatory cells. So that's another kind of scenario. If you remember, T regulatory cells are there for us to regulate the immune response, which way it's going to go. Because by the end of the day, we do give therapy, but again, I just want to emphasize and kind of, push this message that just use of antacids or any other blockers of acids is not a good strategy to deal with gastric ulcers or deal with these kind of problems, because it's an immune problem here, right? So you can see that once the bacteria pass through that, there's immunity that is developing. So we have to basically work for signal transduction pathways. And if you remember me saying, those of you who are interested in drug development and so on and so forth, they need to know the cell biology, and they are all these drugs which are there in the market. And then again, I don't know, the person who was supposed to work on this Helicobacter pylori. I was expecting them to come up with new drugs, but I haven't seen the figure. The figure I was expecting, especially this kind of figure. So this is from the drug development thing, that those people, cell biology, and you can see typically that they want to know what are typical receptors present on H. pylori and what are the corresponding receptors present on your dendritic cell. And how does that signaling take place? And you can see from here that right from the phagocytosis of that particular Helicobacter endosome, they're all signaling pathway that we already talked about. So most of the drugs, as you can see from here, are going to target all of these players over here, all of these players. So this is like new generation of immunological drugs that are coming up to treat such a problem that people used to just take antacid for, or take cimetidine for, or H2 blockers, so on and so forth. So that's pretty much an old story. So you can see from here, once the bacteria gets into the system, and when the message goes to the system, message within the nucleus is whether it's going to produce pro-inflammatory cytokines or beta defenses. And you can see one of the things that this message will go to dendritic cell is to produce IL-18, or it's going to produce IL-1B, IL-1 beta. So you can see both of them will recruit naive T cells, which are cells that have not seen an antigen, and then again will differentiate into two different subsets. So there are two different subsets, and in both cases the responses will go into two different directions. So this is like some of the things for those of you who want to go in that field. We'll try to help the concept forming. How do you diagnose? You have to do microscopy, look for the specimen. I just told you urease test, which is relatively sensitive but highly specific. So what they do is that they ask the patient to breathe into the test, and then urea comes out. It's more of like an alcohol kind of a test, breath test. You can also look for an antigen, which is pretty sensitive and specific, but you have to look that up in stool specimen. You can also culture, but there are problems because you have to culture it in microaerophilic condition. Growth is very slow. The method is insensitive, and then finally you may want to look at the antibodies or serology for an antigen present in your serum for this particular bacteria. What do we do for that? So the established treatment, prevention, and control, other than you would have thought, multiple regimens. There are multiple regimens. We call it a combined therapy with a proton pump inhibitor, omeprazole, very common drug available over the counter. And you can add a macrolide and clarithromycin or a beta-lactam for 2 weeks. So it does show a high success rate. I normally don't ask questions related to treatment, but this is such an important thing. I do ask a question, how do you treat H. pylori? And then again, if you find out that sometimes they do gastroscopy, and they take a culture and they do find that you have Helicobacter pylori. So what they would want you to do is that you want to initiate a prophylactic treatment, and then because the whole idea is that they don't want you to go to that condition where you may have adenocarcinoma of even lower esophagus. They are trying to come up with vaccines, so far not available. And then again, if you have bought the original textbook with a student consult ID, there are quite a few flashcards available and some of the case studies available for you to kind of realize the importance of this case and how do we normally treat them. And I was told by the students working on this particular bacteria that they are quite helpful. So these are like some of the facts that you want to learn or memorize or cram. And you can see from here, gram-negative spirobacilli, virulence factors, all tons of them, diseases, epidemiology, treatment, so on and so forth. So Helicobacter pylori, you must have realized by now, that is an established causative agent of acute and chronic gastritis for gastric ulcer, duodenal ulcer, and then duodenal ulcer is known to form two important cancers. One is adenocarcinoma, and other is a marked lymphoma. Now, in the same set of microorganism, another microorganism they want to discuss today is Campylobacter jejuni. And again, if you look at the structure for that, it says fecal specimen, and you can see curved, thin, curved, gram-negative over here. So you can see over here gram-negative, thin. If you biology, virulence, and disease, gram-negative, and then again, remember the most important slides for all the bacteria, and you will see quite a few questions on that, and I want you to know what is pathogenesis? How does this bacteria tend to cause disease? How do they adhere? How do they move? How do they invade intestinal mucosa? So for Campylobacter pylori, remember I said, alluded to Helicobacter pylori because of the autoantibody production. It is known to cause a disease which is more towards an autoimmunity, where your own cells will go after your own cells and cause disease destruction. Now, the same kind of antigenic cross-reactivity between the oligosaccharides, which are there in this bacterial capsule over here, and then the glycosphingolipids on your neural tissue. Remember these epitopes, they're strange. So they have the same sequence of events on your neural tissue as compared to the bacteria has. So if you make an antibodies to that, antibodies of course will bind to this bacteria, will also bind to your neural tissue. If they do so, you have a problem. That's an autoimmune disease. Very famous Guillain-Barré syndrome. So Guillain-Barré syndrome, very common question asked, again is restricted to Campylobacter jejuni and not Helicobacter pylori. Again, this causes acute enteritis with diarrhea, typical malaise, and the good thing for this is that most of the infections are self-limited, so you don't need to take anything for that. And sometimes if it persists for a longer time, then we may have problem. One problem, though, of many different subtypes of this particular bacteria, Campylobacter fetus, basically is associated with septicemia, so it is dangerous. The bacteria, when it goes into your blood, it has kind of penetrated too far. It's not restricted itself to mucosa, but it's gone into your blood, and wherever the blood takes, so it's more of a disseminated, widely spread out disease that may involve multiple organs. So it's something you don't want. Now, remember I said that we basically are interested in human disease, but some of the diseases are such in animals that affect us, right? So in this case, this is known to be a zoonotic infection. So basically coming from poultry, but again, we all consume either poultry directly or some of the products. Most of the time we acquire that, these are like foodborne diseases, by contaminated food, unpasteurized milk, or contaminated water. Person-to-person spread does not occur normally. And then again, remember, as compared to some other bacteria where you need only 1 or 2 bacteria, for this particular bacteria, in order to establish disease, you have to have very high content of bacteria, right? Or what you can do is that if you take too much of antacid or you mess up your acidity, you will give a chance of this bacteria to cause problem even at lower level, because your acid's not working. It's present everywhere. It's like a typical enteric infection. And if you look at the pathogenesis in a way, and you can see from here, this is a typical, there are several environmental reservoirs. So you can see that it can colonize the chicken gastrointestinal tract, for example, very high number, and they are there in mucosal layer. And then again, it is passed from those animals, you can see from here, to the water, and you can consume the water, or these animals are then eaten up, and you get that infection. So if you look at the mucosa of the chicken, you can see these bugs over here, and then again, they are transferred to you. Or you can also see that they can be also picked up to the water being fed to cows. So it's a multifactorial situation that you have Campylobacter hanging around in environment and ecosystem. So your drinking water gets contaminated. You consume animal product, like in this case, unpasteurized milk or meat, or you consume chicken. Again, these things will add up, and it was going to cause you a problem. And remember, as I said earlier, I gave you kind of an upshot of how powerful that flagella is in terms of poking holes into your cells. And this is for real. So if you see some of the studies which are done recently, and these are, as I said earlier, the slides that I looked upon this morning, you can see a typical structure of this bacteria. They're gram-negative, so of course they have an inner membrane, they have an outer membrane, they have a periplasmic space, and they have a thin peptidoglycan layer, and they have lipooligosaccharide. And this is what I was talking about. So you can see they have very strong, properly propelled, very organized kind of properly propelled a structure that goes out of the capsule and will cause that push and penetration power for not only adhesion, but basically going into the system. So there are quite a few similarities if you pay attention to the human in terms of chicken. And again, you will see that mucosa is common, it goes in waves. In human, we have neutrophil, dendritic cell, macrophages. And of course, in chicken also we have macrophages and some other kind of cells. And the whole problem is that, again, in humans, we have NF-kappa B signaling, if you remember transcription factors. Mostly pro-inflammatory cytokines, where you have inflammation, diarrhea, and clearance, as compared to this one, where basically is a disruption of immune response that these people have. And then again, they basically may have a benign, it's not that kind of a problem. So they can act as a carrier, not do much damage to themselves, but pass it on to the people who consume dairy products. Again, these are some of the mechanisms that are proposed, and how are we going to fight Campylobacter species? Because in order for us to develop a drug, in order for us to come up with especially immunopharmacological drug, we really have to understand the immune response. Again, same set of slides that you have. You have a commensal bacteria. You see tons of commensal bacteria that are out there in the lumen. Then you have mucous membrane. You have and just intestine, because intestine has a higher number of as compared to the gastric mucosa you saw earlier. So Campylobacter goes over here, penetrates itself, and then it can either go transcellular or paracellular. You can see the paracellular, where you have cell-to-cell adhesion molecules like CAMs. So they kind of like opening gates. They can open up and allow this bacteria to go in. Once they go in, then we have all those cells, neutrophils, macrophages, sitting over there, and they should take care of that. And then you have cells of innate immunity or components of innate immunity, like complements and so on and so forth. They will try to go after those bacteria. We really have to understand what role do they play and how we can assist them or enhance the effect that they may have. How do you diagnose? Again, for both of the bacteria that we have discussed so far, stool specimens is insensitive but specific. So you can also culture but need specialized media. You have to give them a microaerophilic, means reduced oxygen. So you have to have elevated temperature. You have to leave it for a couple of days. You can also detect antigen in stools that's moderately sensitive and very specific compared with the culture. So stool specimens, looking for those antigens. How do we treat them? As I said earlier, if you have mild gastroenteritis, infection is self-limited. It's going to go, but you need to make sure that you don't lose your fluid and electrolytes. It may cause severe gastroenteritis and septicemia, and you have to make sure that you have this primary sanitary methods or healthcare methods of washing and making sure the foods are properly cooked, so on and so forth, and also make sure that the public health people are taking control of your water supplies. Because I was reading an article this morning, and it says that if you were to sample your kitchen today, especially the kitchen cloth that you want to wash your utensils with, and the soap and this, everything will have Campylobacter jejuni. There's so much of this thing going on, And there are many other important things that we are still learning from what's going on in terms of establishing hygiene, especially in food industry. Again, I would recommend that you can, maybe it's too late in the game that you want to go and purchase that online version, but those of you who have, you can see some of the case studies are really interesting. This is basically a person has a 10 bloody bowel movements per day. So you can see things can go nasty. Don't think it's a simple, benign thing. 10 bloody bowels a day is too big a problem. Lasting for 1 week, and it has uncooked chicken camping trip. I hope they don't serve them chicken when they go for legislative day in Springfield. You can call them up and tell them that we just found out that be careful. So trigger words are bloody diarrhea, undercooked poultry, thin curved gram negative, and these are some of the essential facts. And as a rule, I'm pretty sure I may have said it many a times, that if the bacteria touch the superficial intestine, it's going to cause irritation, so gastroenteritis. If it goes deeper, then the blood will come. The more deeper, more blood will come. It goes into the vessel, bacteremia. So that tells you the bloody diarrhea. The moment you see bloody diarrhea, you are looking for an invasive kind of bacteria that is going all the way through. And then again, there are flashcards available. You can see some of the information. Flashcards are good sometimes, not all the time, because I don't prepare my question from flashcards, but I can assure you that we have these important things: motility, oxidant positive, virulence factors, anti-complement capsule-like, adhesions, diseases, epidemiology, prevention, and so forth. So this will give you an idea. Now, having said that, what I want you to do for this particular lecture is that, as I said, that I've started making questions for this exam since this morning, but just looking at the lectures that I gave it to you, when I have 3 different bacteria in given one lecture, so I would contrast and compare them, right? So if you have 2 lectures over here, so make sure the questions are not coming from Helicobacter pylori alone. I'm going to compare it with Campylobacter. That's what they are. So make sure you look for familiarities. Many a time people come to me and they say, I know the subject. Well, that's good. I don't deny that. You do know the subject, but you pretty much don't know how to compare and contrast. So regardless of your ability to memorize and cram, you need to know how to compare and contrast, because the questions are such that they have a stem and they have a distractors there. And the words distractors suggest to me similarities. So you have to be very careful, paying attention. Am I talking about aerobes, anaerobes? What am I talking is not. These are some of the things which are little subtle differences, and that will make a major impact if you were to go and pick up a particular choice. Because knowing a subject is totally different than how well you're doing on multiple choice question exam. This is a beast in itself. I can assure you, but the thing is, those people who study less, but they are study smart and they know how to do these questions, they will always perform better, regardless of whatever they do. Because this is how the system works. That's why I've not reached that level, but I'm developing to give you hundreds of questions. You get used to this doing questions. The way to prepare for medical subject is to do the questions. To prepare for the medical reading is not enough. Reading is enough. You want to read it before the lecture, after the lecture, good enough. Once you are done, so you pretty much have a couple of hours. I do my reading. I come from 9 to 11 for this lecture, reading part. And I go back, and maybe for you as well, immediately read it one time. Done away with. Now, after that fact, you need to practice the questions. Because that's where, unfortunately, we don't have any other way of assessing you. But that may answer the question that you may have, that I know the matter. Well, I agree with you. You know the matter. I'm pretty sure you worked hard. But by the end of the day, the assessment still has to be made through multiple choice questions. And that's where it needs practice. So this is the exams. Whenever you study in the exam, this is a study academic thing. It's an education. When you take an exam, it's a skill part. Try to kind of differentiate between that. And many a time, the assessment tools are skill-based. You know, it's just like there's always, in a crime scene, for example, there's always a clue. No matter how good a person is, a criminal is, always find a clue. So even if you don't know anything, for example, you're totally blank, you go in the test, there are always clues there for you to pick up an answer. There's always are, right? Things are common sense. Things are simple concept, but it's usually disguised. It's not like how many fingers are there. It's not like that. 2 hands, 2 eyes. This is not like that. And again, this is a very, very basic information. So that's why my advice, that's why I would never miss a class just because I'm preparing for micro exam. It doesn't make any sense to me. Because no matter what you do from now, you can spend 140 hours reading. It's not going to make any difference on the outcome of that. I would rather go to school normally, attend every classes, gain something, and still do the test. I'm pretty sure there will not be any difference in terms of the output that you have. Okay? So I will stop here, and if